2, 3, 4, 5, 6. All right, ladies, if you have any questions, you can type them into the chat, or you can unmute yourself and just use your audio. There's a little button if you put your cursor over your window where you can see yourself, you can click on unmute, and you can certainly speak to the group if you need to. So remember, all we're doing today, if you weren't with us at 4 o'clock, this is just for questions about lecture material. Oh, that's all right, Sarah. I know there was a problem with the meeting, not the 4 o'clock meeting today, not showing up on our schedule. I secretly am suspicious that Zoom is getting a little overloaded right now with with thousands and thousands of meetings that are happening. Yeah, so if you missed the 4 o'clock, no worries. I'm gonna post the video of the 4 o'clock up on YouTube. I'm posting all our Zoom meetings up on YouTube in case you want to go back. And again, today's just question and answer. Just so you know, the lecture for transcription and translation is up on YouTube, and it's in our playlist now, so you can find it easy. And then after you watch that this evening or tomorrow, you can take a short little lecture quiz for it too. So I know most of you haven't watched the transcription translation one yet, but does anybody have any questions about DNA replication or mutations, that lecture material? Does anybody have any questions about that? I think the replication and transcription and translation lectures are easier than the metabolism lectures. I think metabolism is difficult. I think it's a difficult topic. Hard to sort of wrap your head around. DNA replication and transcription and translation, I think, are a little more straightforward. So remember, we learned some new vocabulary in the replication lecture. We talked about what a gene is, what a genome is, what genotype and phenotype are. Those are words that get used a lot in modern language even. The word genome, I mean, you would never hear the word genome used by anybody but a scientist, even 10 years ago. And now you hear it used much more often as the general public gets more and more comfortable with genetic language. Remember, we talked about the 3 statements of the central dogma. DNA used to make RNA through transcription, RNA used to make protein through translation, and it's protein that creates traits in an organism. One of the challenging parts of learning about genetics is keeping straight which code is which. But the nice thing about DNA replication is that the whole DNA molecule is getting copied. So it's not like transcription where just one strand gets used because one of the two DNA strands has the gene in it. When you talk about replication, DNA replication, both strands get copied and the entire strands get copied. So replication's a little more straightforward than transcription and translation are. Remember, everybody uses the exact same genetic code. It doesn't matter who you are on planet Earth. If you're alive, if you're a cell on planet Earth, you use the exact same genetic code, which is really, really remarkable if you think about it. Remember too that our microbial friends are able to transfer their DNA vertically and horizontally. When they get ready to divide, when a microbial cell, a bacterial cell, for example, gets ready to divide, it has to do DNA replication because it has to copy its DNA. That's vertical transmission. A parent cell creating two daughter cells and passing its DNA down. So from parent to daughter cell. But what makes microbes amazing compared to animals, at least, is that they can pass DNA vertically, horizontally as well. They can pass bits and pieces of their DNA to their neighbors. And that's, from an evolutionary standpoint, that's really useful because that helps keep them diverse. It makes it more likely that different types of microbes, different species of bacteria, for example, will have some unique features to them that other species won't have. It's good to be different from an evolutionary standpoint. You don't want every single bacterial cell to be exactly the same. Horizontal transmission really helps increase the genetic diversity in our microbes. So remember, they can do conjugation. That's the transfer of DNA through a sex pilus. They can do transformation. That's when living cells pick up bits and pieces of DNA from the environment. And they can do transduction, which is a process that involves viruses picking up bits and pieces of bacterial DNA and transferring them from one cell to another. In this lecture, we talked about what DNA looks like structurally. We looked at the subunit of DNA, what we call a nucleotide, and how it's got a sugar in it and a phosphate group and a base. And we said we number the carbons in the sugar because it turns out a couple of these carbons are important for us to understand in terms of where they lie in the DNA molecule. That's the 3' carbon and the 5' carbon. We talked about the idea of complementary binding because, of course, the two strands of DNA are not exact matches of each other. They're complementary to each other. Where there's an A in one strand, there's going to be a T in the other strand. And where there's a G in one strand, there's going to be a C in the other strand. That's complementary base pairing. And remember, we said, while each strand of DNA is built with covalent bonds in between the nucleotides, what's holding the two strands together is hydrogen bonding. What holds the A's and the T's together is hydrogen bonding. What holds the G's and the C's is hydrogen bonding. So we often talk about the sugar phosphate backbone of DNA. the two strands have that backbone in them, that covalently bonded backbone. And in the drawing, remember, that's the big, thick red arrows. And the bases are sort of jutting off of each strand, and then they hydrogen bond with each other. And then that whole double-stranded structure twists into a double helix. We talked about how DNA replication is called semi-conservative. And remember, all that means is that each of the individual strands is conserved. In other words, it's used as a template. Each of the individual strands of DNA is used to build new DNA onto. That's what we call semi-conservative replication. We use each individual strand as a template. We don't use the whole double helix as a template. We take the strands apart in order to copy them. And we talked about the enzyme that does this very important work. And this is an enzyme called DNA polymerase. Remember, there are lots of enzymes involved in replication. There's all the different steps of replication have a different enzyme doing them. But the big, most important enzyme, you could argue, is DNA polymerase, because that's the enzyme that brings in each new nucleotide and builds the new strand of DNA on top of the old strand. We said that enzyme can only work in one direction. It can only build DNA in the 5' to 3' direction. And we said that what that means is it can only add new nucleotides onto the 3' carbon. So it can only make DNA in one direction. It can only add new nucleotides, new bases, onto the end where the 3' carbon is. And that's important because, remember, the two strands of DNA are anti-parallel to each other. That's the other term we talked about. One strand is oriented in one direction and one strand is oriented in the other direction. Going back to this slide, if you look at the red arrows, this strand has its 5' end over here and its 3' end over here. And then if you go over here, this strand is oriented in the other direction. They are anti-parallel to each other. And the reason we put a little arrow down here at the 3' end is because we're reminding ourselves that this is the direction that the strand will grow in when it gets replicated. We're going to add new nucleotides only to that 3' end. So this strand is going to grow in this direction, and the other strand would grow in the other direction. Remember, there's a point of origin, or what you'll also read in some textbooks is called the origin of replication. This is just the exact point where the double-stranded DNA molecule gets separated. And it creates what we call a replication bubble. It creates an opening. And you can see that on this slide down at the bottom. It creates a little bubble where the two strands have come apart. And the bubble just keeps getting bigger. We open it up at this end and we open it up at this end. And each one of the old strands gets copied. We build new DNA on each one of these old strands. Remember that the cell has to lay down a primer, a little short piece of RNA on top of the old DNA to get things started. That's probably one of the strangest aspects of DNA replication. And we looked at this little drawing of the circular chromosome. We said this is the If this is where the origin of replication is, we could see just a little tiny replication bubble here. That replication bubble is going to get bigger and bigger and bigger over time as the DNA gets copied, as it gets replicated. New DNA is shown in red. Old DNA is shown in gray. Replication goes in both directions around that circle. One of the challenges when you're learning about DNA replication is this concept that some of the new DNA is made continuously and some of it is made in fragments. And I think I said in the lecture not to worry too much about this, the details of this. But I want you to understand that because DNA can only be made in one direction, it means that on part of the new strand, of each new strand, part of the strand gets made in what we call leading strand synthesis. That's continuous. And part of it gets made in lagging strand synthesis. That's discontinuous or fragmented. And of course, as we always do, the guy who discovered this got his name on it. So Dr. Okazaki got his name forever immortalized. We call the pieces, the fragments, Okazaki fragments. So in this drawing on this slide, you can see the replication bubble. And remember, on one old strand, the top black strand, that's one of the old strands, the new DNA can only be made in one direction. So that means that from the origin, part of it gets made continuously, and part of it gets made discontinuously. Part of it gets made as one long piece, part of it gets made in fragments. On the opposite strand, we're making it in the other direction because the strands are anti-parallel. Part of it is being made continuously, leading strand synthesis. Part of it is being made in fragments, lagging strand synthesis. Just something to know about replication. And remember, at the end of all of this, when both of the DNA strands have been copied completely all the way around that circular chromosome, the cell still has a lot of work to do. The cell has to join together all of these fragments and make one piece, and then it has to join that one piece to this one piece so that you end up, again, with a complete strand of DNA. And there's all kinds of different enzymes that are going to do that work. In the end, we need to have 2 DNA molecules. Each one of them is going to have one old strand in it and one new strand in it. Semiconservative replication. And at the end of the lecture, I put in this summary slide, or the 2 summary slides. Remember, there's different enzymes for each different step. We don't need to know the names of all those enzymes, but we do need to know the name of that key enzyme, DNA polymerase. This is the enzyme that is actually doing the building of the DNA. So you have to open up the double helix. You have to put a primer down, and then the DNA polymerase enzyme can come in and build DNA. And the way it knows how to do that is by using the old DNA. It builds a complementary strand. So it essentially reads the old DNA. If there's a G in the old DNA, the enzyme lays down a C. If there's a T in the old DNA, the enzyme lays down an A. And it just keeps doing that all the way along the length of that old strand. And then, as we said, the primers have to get removed. The Okazaki fragments have to get joined together. Everything has to be seamlessly attached. So you end up with 2 molecules of DNA by the end. And each one has one old strand and one new strand in it. Miss Millie. So we said that in this lecture, we pointed out that this enzyme, this DNA polymerase enzyme, is actually really amazing because it works really fast, number one, and it is essentially very accurate, but it does make mistakes. Remember, we said about 1 every 100 to 1,000 bases in the old DNA, the enzyme puts the wrong base in. And the good news is that there are systems in the cell. There are systems in the cell to check for errors and to fix errors. And most of the errors that the enzyme makes get fixed. And we saw that in the ultraviolet lab, if you remember. She's like a big baby. She weighs like 50 pounds and she thinks she's a lap dog. I know. So, you know, when we think about mutations in our DNA, a lot of us automatically think about chemicals, you know, getting exposed to chemicals or getting exposed to radiation. And those things can cause mutations, but copying DNA causes mutations too. So anytime a cell is rapidly dividing, we see more mutations in those cells because the enzyme makes mistakes sometimes. And the good news is that most of the mistakes get fixed, but some of them don't. And those are what become mutations. If you think about the virus right now, the virus that is causing COVID-19, right now, and of course, viruses don't replicate their own DNA. They let a cell do that. And COVID-19 virus, as it turns out, doesn't even have DNA in it. It has RNA in it. But the idea is the same. All of the humans that are infected with that virus right now are copying its genetic material. Think about how many times that viral genetic material is being copied in this moment. Okay, that's how viruses mutate. They mutate a lot because they get copied a lot when humans pass them around. And the good news for us is that the data right now is showing us that it doesn't look like it's mutating very much. That's good news. Because boy, there's a lot of people who are replicating that virus right now in their bodies. So it's good news for us that it's not mutating any faster than it is. That'll make it easier for us to build a vaccine against it. So mutations, we said the 3 biggest causes of mutations in DNA are errors during DNA replication, exposure to chemicals, and exposure to radiation. Those are the 3 biggies. And the reason we care about mutations, which are just permanent changes in the order of the bases in our DNA or in microbial DNA, is because mutations can lead to new traits. Mutations lead to changes in proteins sometimes, not always, but sometimes. Remember, we said some mutations are silent, but some mutations change the order of the amino acids in proteins. And when you change the order of the amino acid, you're going to change the function of that protein. Some of the mutations are very bad. Some mutations cause disease, right? A lot of the horrible, especially inherited diseases, are caused by mutations in the DNA. But mutations are also the cause of evolution. Mutations create good things too. The reason that organisms mutate over time, the reason that evolution occurs, is because new traits come along every once in a while. And those new traits come about because of mutation. So it can be good and it can be bad. And we care about it a lot in our microbes because sometimes microbes mutate and they become more pathogenic. And that's exactly what happened with this COVID virus. It mutated. It used to live in bats. It used to infect bats. And it mutated and it gained a new trait. And that new trait was the ability not just to infect humans, but to infect the cells very deep down in the lungs of humans. And that's bad for us, right? That's a bad trait for a virus to have, for our health anyway. So some are good, some are bad. And we said we typically discuss mutations, remember, as either being point mutations, that's a base-to-base mutation, or chromosome level mutations. We don't talk about chromosome level mutations in microbes because they only have one chromosome. We just don't see them the way we see them in humans. Some of the worst cancers, for example, that we can trace back to a mutation are chromosome level mutations. But point mutations can be just as devastating. So we talked about what a substitution mutation is, what a silent mutation is, what a nonsense mutation is, frame shift. These are just changes in a base through a mutation. So remember, if you're changing one base for another, that's a substitution. Sometimes those substitution mutations are silent. And what that means is, even though there's been a change in the DNA, there's been a change in the order of the bases in the DNA. That's what a mutation is. Sometimes that change doesn't really matter because you've changed the codon in a way that doesn't change which amino acid goes in that location in the protein. That's a silent mutation. You would never know you had it. Other times, substitution mutations do lead to changes in the order of the amino acids, and then you get changes in the protein and changes in the trait. Nonsense mutations are a type of substitution mutation. We separate those out because the substitution mutation creates a stop codon where there didn't used to be one. So that's going to create a big problem because the protein isn't going to get made as long as it should be. The ribosome is going to hit this brand new stop codon in the middle of the mRNA somewhere, and the protein is going to end up being much shorter than it should be in terms of the amino acids. And then a frame shift, remember, is different from a substitution. We're not just swapping one base for another. We're either adding in an extra base or we're taking out a base completely. Frame shift mutations are also pretty devastating in that you get a big change in the protein that comes from that. So somebody asked about the COVID virus. Do we think it will go to dogs or to other animals? There was a report, there's been a couple of reports out of China, and I'll say this, take everything that you read about coming out of China with a grain of salt, because we get very limited information from China. There was a report several weeks ago about a dog who tested positive for the COVID-19 virus. Now, from everything that we've been able to gather from the Chinese scientists, and it hasn't been much, what they discovered was the presence of the viral particle in the dog's nose. Now, that's very different from the dog having the disease caused by the virus. Do you see what I'm saying? There's a big difference between the presence of the virus being in an animal and the virus being able to cause disease in that animal. So right now, it doesn't look like other organisms are susceptible to this virus. That may change, but right now, at least, we are the host. Remember when we talked about viruses, we said viruses are host specific. They have one host at a time. Now, that can change over time as they mutate. They can jump from one host to another to another. That's what happened to this virus. It jumped from bat host to human host. It's uncommon, though, for a human virus to jump to dogs. It's very uncommon. I can't think of a single example. It's much more common for bats and humans to send viruses back and forth. And remember, the other animals we talked about were pigs and birds. Pigs and birds and bats are the biggest sort of reservoir of viruses for us because we're genetically just closer to them in regards to our receptors. Good question. Yeah, I haven't heard yet. I don't know yet that we know what's happening at the Bronx Zoo about that tiger. We're going to have to wait and see. We're going to have to run some tests to see if that is truly an infection in that cat, or the other question is it the COVID-19 coronavirus or is it a different coronavirus? So we have to do a little more testing. So the other thing we talked about when we talked about DNA replication was we talked about how we can do it in the laboratory. And we do it for our own purposes in research and medicine by using this technique called PCR, polymerase chain reaction. We talked about the steps of PCR and what happens at each step. We talked about the special kind of DNA polymerase that we use for that reaction and how we can make millions of copies of DNA in a test tube in a relatively short amount of time. One of the tests that we're using for COVID-19 virus right now is PCR. So the test that the Centers for Disease Control is using is a PCR test. So another way of thinking of that is we are looking for the genetic material in the virus. And if it's there, we copy it 1 million times so we can see it in the test tube, essentially. That's the test for the virus is a test for its genetic material, its RNA strand. And we can use PCR to find it. Now, there's another test that's coming up now that we're hoping we will be able to distribute to more people, and that's an antibody test. So there's a very different kind of test that looks for whether or not a person's immune system has responded to the virus by producing antibodies. That test is much easier to do than PCR. That test is cheaper and it's faster. So we may be hearing more and more about using antibody testing as we go forward. All right. So as you look at the transcription and translation slides and you take that little quiz over the next day or so, if you have any questions about that material, remember you can post them on the discussion board and I'll be checking and answering questions as needed so that you'll be ready on Wednesday for the exam. Remember, you're free to use your notes during the exam on Wednesday. I would recommend, though, that you go ahead just like normal and make yourself a card. I would do that. I would go through the exercise of making yourself a 3x5 card, even though you can use all of your notes, because making the card is helping you study. Making the card, choosing what information to put on that card is actually helping you study. And you'll be able to find the information faster during the exam. So if I were taking the exam on Wednesday, that's what I would do. I would study just like I was going to take it in the classroom, and I would make my little card, because that helps you process the material as you're studying it. The exam is timed. You can go past the stop time, but if you go too far past it, you'll lose a couple points. The reason it's timed is because we want to encourage you to learn the material and not just, look at your notes and answer the questions. So I'm really encouraging you to prepare as though you were taking the exam in the classroom. You'll have plenty of time if you're prepared, if you've studied. And as we go forward, if anybody has any other questions about what's going on with the COVID-19 virus, you can also post those questions on the discussion board. I'm happy to have us talk about that as we go. Any other questions? All right. Well, I'm going to let you go then. I'm going to let you go and take a look at those slides if you have a chance. And I'm going to go and see if I can get Millie to stop barking. So keep in touch with me if you have any questions, okay? And otherwise, good luck on the exam on Wednesday. And I will see you after Wednesday afternoon in lab. All right. We'll see you then.